Tell me when. Okay. Okay, everyone, it's a pleasure to introduce David Schulz to, I guess, the few of you who may not know him. It's probably not that many. Dave did both, I think, undergrad research and his PhD with Gene Robinson at the University of Illinois, Urbana-Champaign, or as Dave calls it, Banana Shampoo. He then had a postdoc lined up actually with Ron Harris Warwick, but his wife's postdoc opportunity at Cornell fell through and they both ended up in Boston instead. So Dave joined Eve's lab, where he and I overlapped for a few years. In Eve's lab, Dave did something sort of heroic and maybe also mildly terrifying. He had done lots of behavioral stuff, I think, and some biochemistry in Gene Robinson lab, and I'm assuming in that lab, you're also somewhat familiar with behavioral genetics, but his task as a postdoc then was to both learn electrophysiology and biophysics and things like that while establishing qPCR and related molecular work, which he himself had very little experience with at the time, and there were a few people around he could ask anything from. So his plan was to see if the individual variability of gene expression tracks with the variability of ionic currents. And now, ionic currents and conductances are a very indirect measure of channel protein expression to begin with, and there was no real a priori reason to assume that mRNA expression of channel genes correlates well with currents, but luckily they absolutely did. And Dave, together with Jean-Marc Goyard, produced these papers that sort of unequivocally confirmed earlier work from Jorge Golowash and others in Eve's lab, that the substantial inter-individual variability of ion channels is real. And they also found that cell type specific correlations between different channels, potassium channels mostly, are reflected at the gene expression level. And a number of people have grappled with that kind of thing since. So these are certainly among my favorite SDG papers ever, actually. He then started his own lab at the University of Missouri. There he continued some of the simple circuit stuff with the STG and also the cardiac ganglion. And so far has only partly defected to spinal work. But he continues to combine physiology and gene expression studies there. He's been very productive, and one of the things I admire most about Dave is that he always does everything from a functional or physiological perspective. And I think it's very easy to actually lose sight of that while wading through the complexity of genomic data. So today he will tell us about what I assume are both the rewarding and the painful sides of these approaches, and take it away, Dave.
Great, thank you so much, Dirk. Everybody has my slide, I assume, the title slide. The sharing works. Thank you for the introduction. That was incredibly generous of you. Thank you for letting me speak in this series. Unlike my more esteemed predecessors, I know that I have too many slides, but I'm not going to ask if I can keep going. I'm just going to keep blasting through. So stick with me, and I will do my best not to go over. But you know what these talks are like. It's hard to encapsulate. And so what I really wanted to, as I struggled with how to put this talk here together, particularly for this group that knows physiology so incredibly well, and has been generous enough to let molecular biology sort of infiltrate the holy sanctum of membrane biophysics and neuromodulation. What I really decided I would do is, and let me get my laser pointer set up here. We'll go with blue if that's okay with everybody. What I decided I would do is basically focus on what I think are three main contributions of molecular biology to STG circuit physiology. And these are using molecular approaches for gene discovery, for molecular profiling of cell identity, and then the functional insights that we might be able to gain. I just want to say up front, with all apologies, that this is entirely selfishly an STG-centric presentation. For folks like Paul Katz in the room, my apologies that there's only STG molecular biology in here, even all the beautiful work that you're doing in our favorite sister taxa. For even other crustacean folks like Tim McClintock and Charles Derby and such, this is really STG centric. And so I won't be referring to any of that work, but all of that informs the work that we do. And then I ultimately decided that what I'd like to do is take a more didactic approach to this. You know the literature, you've read the literature. I wanted to give you sort of my insights of of how I think this approach can contribute to circuit physiology rather than just a laundry list of the papers that have been published in this area. And so I hope you'll appreciate or at least forgive sort of the more didactic perspective that I'm taking on this. And then finally, I feel like I am sort of obliged to do some very, very simple background. And so this is one of those insult your intelligence slides to remind you that when we talk about gene expression, we are talking about the process of going from genetically encoded material in the nucleus through the processes of transcription, transcriptional post-processing, trafficking, translation, and then ultimately mature protein. But there's no reason to linger on a slide as overly simplistic as this, because you know this is far more complex a process, and this is barely scratching the surface of all of the regulation of gene expression that takes place in the nucleus with transcriptional regulation, the processes of transcription that produce an mRNA copy of information encoded in genes, the processing of splicing, polyadenylation and capping, the trafficking of this out into the nucleus. And then it turns out that RNA isn't the end, even though that's where I tend to finish, but rather I guess there's some other stuff that might happen before something happens physiologically. And this slide is to both appreciate that complexity and point out that we're going to focus almost exclusively at this level here, sort of that mature steady state mRNA point where we're quantifying these transcripts in this state and a nod towards degradation pathways that play a role in this as well. And so when I talk about gene expression, I'm talking about how we can measure these things. And so really, again, briefly, gene expression is the process all the way from the initiation of transcription to mature protein. And really, when we think about the transcriptional event and we reach a steady state point, there are really, 4 tried and true methods for looking at mRNA levels. We'll touch on qPCR and RNA-seq today. If you're still doing northern blots, I'd like to meet you. That's fascinating. And microarrays are also largely only the domain now of sort of carryovers of some human arrays and such for sort of historical purposes and the fact that they work and they're FDA approved in some contexts, but RNA-seq has largely supplanted experimental microarray work as well. If I slip and I say gene expression, I'm really probably, I'm really probably talking about something at the level of mRNA measurement, although expression is really this whole process. And so with that as my really, really brief introduction, I want to get into these 3 levels of where we're potentially making contributions. And so the first is really the use of molecular approaches towards gene discovery in these, for lack of a good term, this is a lousy term, these non-model systems. Now, I don't appreciate that terminology, but we'll just say that these are less genomically developed systems. And luckily, technology is making that less and less an issue. And so really when I'm talking about gene discovery, I'm talking about the use of sequencing approaches to identify predicted genes that would be interesting to you for STG function. And we're doing this through a process that is sequence homology. That is, how does a known sequence or a new sequence in crab or lobster compare with what's known in other species? Gene discovery really can go forward in 2 ways. One is a candidate gene approach where you know what you're looking for. You take mRNA from your tissue of interest, reverse transcribe this into DNA, and then use some form of targeted specific primers that are designed usually from homology in other species to PCR amplify what you hope is your gene of interest, participate in traditional Sanger sequencing, and then use bioinformatic approaches to identify that sequence as whether it's a potential homolog of the gene you're interested in. And then obviously far more high throughput these days is using RNA-seq to do whole transcriptome assemblies. And we'll talk a little bit more about this in a moment, where we do tremendous amounts of high throughput sequencing, push this into bioinformatic platforms that look for overlapping sequence to make what are called contigs or continuous sequences take the whole mess of those and use search algorithms to some sort of reference sequence or reference genome to identify as many transcripts as possible. Again, at the end here, what we want is our species-specific sequence for our genes of interest. And so we've seen this approach used successfully in the STG. So for example, Sanal in Eve's lab was targeting gap junction proteins, gap junction genes, the annexins in both Homaris and Cancer borealis. She was able to use targeted gene approaches to identify at least 6 annexin genes from these 2 species and look at their expression in different tissues, look at their expression in STG neurons and their relative amounts and correlations. Another really, I think, beautiful study that I was lucky to be a part of was Veronica's study in Dirk's lab looking at CCAP receptor. And I'll bring this back throughout the talk as an example of how we can use different levels of these analyses in STG. But Veronica targeted the Cancer borealis CCAP receptor, was able to target this with an RT-PCR approach, derive a sequence that we felt looked quite a lot through sequence homology to other CCAP receptors, and ultimately look at the expression of this in the STG and in identified neurons of the STG. So 2 really good examples of knowing what you're looking for, delving into crab or lobster mRNA pools and pulling out sequence of interest. More recently, obviously, we've been using RNA-seq in order to generate a high throughput data for gene discovery. And so again, briefly, if you're not familiar with the way RNA-seq works, a very typical workflow is for you to extract RNA from cell populations, tissues of interest. Usually we're selecting out the mRNA because we're interested in coding genes, although it's possible also to size select for small regulatory RNAs, or if you were a ribosome person, you could certainly try to enrich ribosomal RNA. That large collected pool of mRNAs becomes fragmented because the sequencing looks at small sequences, converted to a DNA form through reverse transcription. A library is constructed where those fragments have adapters ligated to them. These are additional DNA sequences that are critical to the sequencing process. And that library is put through a high throughput sequencing workflow, usually on an Illumina platform, although there are lots of platforms these days, which gives you tons of sequence that are bioinformatically assembled into those contigs. I'm really sorry for all the jargon, but this is how these folks talk. These contigs are then looked at in a reference and ultimately the best attempt to identify these takes place. What this then looks like if you're a crustacean biologist is as the work that Adam Northcutt did in our lab in collaboration with Eve to generate a C. borealis reference transcriptome. And so what Adam did is he took mixed nervous system tissue from crab, including brain and the whole STNS and cardiac ganglion STNS, including OGs, COGs, you name it. And we tried to get sort of a whole representative nervous system transcriptome, isolated that RNA, and we polyase selected that. And we did all our due diligence of looking at RNA quality with bioanalyzer, constructed a library, constructed that library. We PCR enriched that library, which simply means we boosted the copy number by taking advantage of these adapters, repurified, enriched, QC'd that, and pushed that into this sequencing workflow. So a whole bunch of gobbledygook for anybody who's interested. But at the end of the day, what you end up with is I think the technical term would be a boatload of sequence that you ultimately try to look for genes of interest that float your boat. So for me, I've always been really biased towards ion channels and membrane physiology. Oh, and this is my note to remind myself that we did this entire workflow in parallel with Homaris. And so we have Homaris and Cancer borealis reference transcriptomes out there and deposited on GenBank. And they've been used very effectively by other groups, as you'll see in a moment, to generate some really interesting gene discovery. I've always been a channel person since I was sort of welcomed into the STG fold. And so I asked Adam first and foremost to try to identify every voltage dependent ion channel he could from this transcriptome. And so we've come up with what we think is a relatively comprehensive set of all voltage dependent ion channels. He pulled out TRP channels. We could not possibly escape homage to Ron Harris' work and all the other amine modulation labs and ignore the biogenic amine receptors. And so thanks to all of Deb Barrow's hard work on identifying biogenic amine receptors as well, we had a lot of good sequence to work with to identify these. We've been working really hard to figure out what's the deal with glutamate receptors in crustaceans and in STG, and we started by pulling out all these receptors. And then looking at things like nicotinic and muscarinic as well as GABA receptors. And so this is my way of sort of presenting to you that this is post hoc RNA-seq gene discovery, where we hand curated each of these sequences with as much care as we possibly could. Luckily for a lot of the folks in this crowd, we have talented folks like Andy Christie and Patsy who really like to think about peptide modulation and transcriptomics. And so recently Patsy and Andy published a paper where they used the C. borealis transcriptome to pull out putative neuropeptide receptors. And so here's a figure from their paper trying to identify as many neuropeptide receptors as possible. And also the peptide precursor genes themselves. And by the way, throughout this talk, because it's recorded, I know this is small and hard to follow, but I've tried to provide really sort of thorough citations so that you can go back and look at this work and be able to mine these data for yourself, because there's a lot of really beautiful stuff going on in here. And we can go on and on in terms of how we can use transcriptomics for gene discovery, but I figure these are sort of the two major families of things that the STG world cares a great deal about. Now, I promised in the title, though, that there would be limitations associated with these approaches as well. And I will say that even though this is an absolutely critical first step in really new species-specific molecular work, this gene discovery by homology has real serious caveats to it, because it's all done through sort of algorithmic search approaches. One of the workhorses of this is an algorithm called BLAST, which is the basic local alignment search tool. And the way BLAST works is it tries to align sequences. This happens to be nucleotide sequence, but it works as well for protein sequence. And it assigns a score to identical matches, to mismatches, and then gaps, and ultimately comes up with a summation score that gives you some indication of how similar two sequences are to one another. The problem with this is that it's completely agnostic to function, and it is incredibly pervasive in terms of wanting to find an answer. And so depending on where you set the parameter cutoffs for these algorithms, it's going to show you sequences that are similar to your sequence of interest, but without a lot of really, I think, extensive care and curation, you can ultimately end up in a game of telephone, where realizing that the other thing that's really prevalent in this is human curation. If 30 years ago somebody identified a gene as X, then without a lot of functional annotation, for example, then over the years, the more and more sequences that are compared to that will start to adopt that name. We might ultimately have sort of a house of cards in terms of gene identification. And I hope this will work, and I hope this isn't too annoying, but I'm going to switch my share if I can, and show you what I'm talking about here. So actually going back to this, I know a lot of the people in the room would be really, really excited to think about a proctolin receptor in Cancer borealis. And so here is where that sequence fell out in Andy's analysis of this. And I also followed up this analysis, or had done this preliminary as well, and come up with the same potential transcript. Now, pardon me for this, and if this experiment fails, you have my apologies. Hopefully you're now seeing a browser window. The other thing I want to tell you about is how beautifully curated all of this work is. So if we're not seeing a browser window. You're not seeing a browser window?
No. I'm
Okay,
not.
let me do this again. Now you are, right?
Yes.
Excellent. Thank you, Paul.
Actually,
And so,
yeah, it's a browser window.
excellent. And so now if we go to Patsy and Andy's really lovely paper here, we can scroll down. We can find all of the work that they've done, and every one of those curated sequences has now what's associated with it called an accession number, which is where you'll find this in public databases. We can find that proctolin receptor and just copy that. If I go to NCBI, National Center for Biotechnology Information, and put that search number in there, not only will it come up with the sequences that they've deposited, but I can very quickly do some BLAST searches myself just by clicking the BLAST button. So I won't go through all these parameters, but if anybody offline wants to go through this stuff and work with this, let me know. We would actually do a BLASTX here because we want to look at translation. And so if I limit this to what is the closest possible ortholog in Drosophila, and I've done this BLAST for you already, here's a list of results that come up. So we get a peptide receptor that's a GPCR, and right there, the second hit is proctolin receptor. We're excited about that. We may have identified a proctolin receptor. But you'll notice as we start going down the list, there's a lot of uncertainty even with Drosophila as to what this might be. So we get a reasonable hit for this FMRF receptor as well. And if we look at some of the alignments of these sequences that are provided, here's that alignment with the proctolin receptor. You can see a very small stretch of this was selected for the outcome. And as you scroll along here, you'll see that there are some conservative substitutions, some overlap, but not an overwhelming amount of sequence information here that's giving you a lot of confidence here. And if I were to go back and blast this against all non-redundant sequences, what I would find is that putatively identified proctolin receptor actually looks a hell of a lot like, more like FMRF amide receptors from all of these species, which are probably named for something that is not functionally identified. So a tremendous amount of care has to be taken here in order to make sure that these algorithms spit out meaningful sequence information. And again, just to be clear, that is, are we back to my slides? Thank you. To be clear, that is not an indictment of the work. If you read Patsy and Andy's beautiful paper, they will spell this out very, very specifically that for proctolin receptor, which they know is a serious target here, there are some mismatches there. But you can imagine 10 years from now, somebody just going to that table, not bothering to read that work and saying, oh, great, I've found a proctolin receptor, build a PhD thesis around it and be very disappointed. And so that is the limitation of letting algorithms, especially when you're dealing with 20,000 genes at a time, take over for really, really serious hand curation of these. And so that leaves us with this term putative. A lot of these transcripts in these databases are labeled putative, as in, this is our best guess, but if we're wrong, it's not our problem. I think that's the translation of putative. And so then how do you get over this limitation and actually start thinking about real functional gene identification? And so I think one of the most beautiful examples of this is work done in Ron's lab by Deb Barrow, where instead of simply being happy that they were able to identify shAL, an A-type channel with RT-PCR, Deb actually went through the trouble to characterize the shAL transcript current by injecting this in Xenopus oocytes, generating really, really lovely activation inactivation curves that clearly identify this as a transient potassium current, finding a reversal potential, shifting that reversal by changing concentrations of potassium using pharmacology, and even going so far in her own hands to compare the fly shAL channel, which was the initial indicator, to the lobster channel, and really de-orphanize or truly functionally implicate her shAL transcript as that underlies the A-type current. This is really gold standard work for going beyond just sequence identification and really talking about functional identification. I love to go back and read all of Deb and Ron's early molecular work because it's some of the most thorough and exceptionally well done work that I've come across. If you're not so lucky as to be working with a protein that would be amenable to an ectopic expression system, then you can do what Veronica did, which is use other functional data to sort of at least make strong inference about function. So for example, with that CCAP receptor, what Veronica was able to do is draw on the work of Andy Swenson and know which STG neurons are sensitive to CCAP modulation, and the work of Mikey's lab and other gastric labs as to which gastric neurons are sensitive to CCAP modulation. And those that are physiologically known to respond to this peptide are in bold. And you'll notice that as Veronica did single cell qPCR, she was able to quantify transcript in those cell types. In those we know are not sensitive to CCAP, again, thanks to the body of work, including Andy's work here, we saw no expression. And you can see this, this is a very well borne out by the known physiology. And so if the gold standard is not available to you, and by the way, expressing peptide receptors ectopically in other like oocytes or cell culture is incredibly challenging. I know Veronica hammered away at this for a while. If that's not accessible, bringing in other physiological data, and here's just a repeat of that Adam Northcutt did that is, with my apologies, not arranged in the same order, but largely recapitulates this result, is at least going beyond simple homology and using known physiology to make real strong inference about that gene identity. And so this gene discovery process is where all of these molecular projects start that have functional insights that I'll get into a little bit. And quite frankly, I find it a lot of fun. It's sort of one of those relaxing activities that nerds do, where you're looking at gene homology, sort of hand curating these and updating databases on the weekends when you've got nothing better to do. So I'm going to go ahead and press ahead into my second section here, and that is using molecular approaches in something that I've really been interested in trying to leverage STG for in terms of functional cell identity. And so really what I've had a fever dream about for the past, I don't know, 10, 15 years at least, is using molecular profiling to really ask, you know, which neurons in a network are distinct at the level of whole cell gene expression. Is the functional identity we see in STG found at the level of transcriptome, at the level of gene regulation? And really the question we're asking here are there unique messenger RNA profiles, cellular transcriptomes, that underlie cell identity and those specific contributions that our cells make to networks? And I think an equally plausible alternative hypothesis here is that perhaps the cell identity and functional output is post-transcriptional, so that there is a baseline of expression that makes an STG cell an STG cell, but what specializes them is translational regulation and post-translational modification and insertion and such. And so I've been sort of trying to track this down, and this has been my white whale for a little bit. And so I got really emotionally invested in this project, which is always a mistake, but I really, really wanted this to sort of be the identification of sort of true cell identity. And so this is work done again in collaboration with Eve, led by Adam Northcutt in my lab, who did that de novo transcriptome assembly. And then as a group, we collected identified STG neurons from 12 crabs, extracted those RNA, reverse transcribed those, working in part with Hans Hoffman's lab at University of Texas. We generated sequencing libraries and did single cell sequencing. Here's some details for the sequencing wonks in the audience. After we got all of those sequences back, we mapped them to our reference transcriptome to get quantitative information associated with expression in each of these cells, and then used a bevy of analytical tools like hierarchical clustering, differential expression analysis, and such to try to track this down. Again, just to put this into graphical format, we took RNA from these cells, isolated them, generated those libraries, sequenced the hell out of them, and generated millions of reads from each individual cell. And then how do you go from that to quantitative information? Well, all of those small sequencing reads that were fragmented are sequenced, and then using bioinformatic tools, they are mapped to where they match the reference transcriptome. And the more often a small read associated with any given gene is mapped, it's counted. And so we would expect, if relative abundance through this process is maintained, more abundantly expressed genes will be mapped to their references more often, and lower abundance genes, like the purple one here, for example, has fewer reads mapped to it. And so we can get quantitative information that is either relative or absolute, or relatively absolute, as I like to refer to it, but depending on what sort of upfront processing and controls we do. So we're now getting quantitative sequence information for whole transcriptome from these individual cells. One of the main tools involved here is hierarchical clustering, and this is an awfully cartoonish way to think about this, but if you haven't thought about hierarchical clustering in a while, I just wanted to briefly explain what this is, these approaches are doing. And so if we had two genes quantified for a whole bunch of different cells, and they fell out in this XY coordinate, what we might start to do, what the clustering algorithms will start to do, is identify pairs that are most close in that two-dimensional space. And in identifying the closest through various distance metrics, you can start to build essentially a phylogenetic tree or a clustering tree. And so those two are closest, and so they're clustered together. We identify another that's just a little bit, and that gets clustered together. The clustering continues until we now start to do higher level nodal clustering of groups of genes that gets added to this tree, until ultimately you build an overall clustering analysis that tells you how similar expression patterns are amongst individual cells going into this. Now, this cartoonish example is a two-dimensional example. We could certainly think about this in multi-dimensional space. And in fact, when we started, what we got was 27,800 some odd unique contigs. And so my PowerPoint skills do not allow me to plot in 27,000 dimensional space. And so I've left this here two-dimensional, but this same idea of clustering is happening across this n-dimensional space. And so ultimately what we're left with then is, can we come up with distinct clusters or nodes that recapitulate known cell identity? And so remember, we're working with LP, PD, GM, and VD neurons. And when we first started this, we just pushed in all possible transcripts from the neural transcriptome of crab. What you're seeing are heat maps that show you the relative abundance, and there are 27,000 rows in this plot. And so relative abundance for a given gene is represented in the x-axis, and each column represents one cell and its entire transcriptome for those 27,000. And so this lets us ask, at the whole transcriptome level, is there a molecular signature that we can identify in this way for these 4 cells? And there's a lot of information in here. And so when we sort of de-anonymize each of these cells in these clusters, we can see that on this whole possible transcriptome level, we get very, very poor cell identification or recapitulation. Basically, this fails to recapitulate functional cell identity. But this doesn't worry me, because there is a whole lot of stuff in here, remember this is whole nervous system, that isn't even expressed. And so we then decided, okay, we'll look at only the transcripts that are detected in at least one of these cell types. That got us down to about 17,000 transcripts that showed up in at least one of these cells. And now this was starting to really get my attention, because you can see we really have started to identify 4 distinct nodes that might start to recapitulate our 4 distinct cell types. So I'm on the path of the white whale here. And when we de-anonymize this, though, you can see that, again, we have completely, for the most part, failed to recapitulate cell identity in this regard by looking at everything that's expressed just in STG. Now, there's a lot of noise in here. And so we decided then to look at the transcripts that are detected in all of these cell types. So maybe what we need is sort of the really commonly expressed stuff across all cell types. And again, we've got 4 nice clusters that has me finally have my aha moment. And again, this completely fails to recapitulate what we know as STGologists is cell identity or functional cell identity. I'm not going to lie to you, I started to get a little bit frustrated at this point because I really thought that we were onto something with this functional genomics. And so after sort of dusting myself off of this analysis, we decided, okay, well, let's use what tools are at our disposal. And so what else we can do is look at what's differentially expressed across these cells. So ask, okay, get rid of everything that is basically common and look at what's really truly differentially expressed that we can detect here. I won't go into too much of the analysis, but when we did pairwise comparisons, we ultimately ended up at the criteria we set of sort of a 0.05 cutoff and at least 2-fold or higher expression difference. 171 of these transcripts that were differential between PD and VD, 187 between PD and LP, and 87 between LP and VD. And those are vanishingly small numbers, quite frankly. And ultimately, if we now take what was our all expressed contigs, everything possible from this, and what's differentially expressed, you can see now when we look at what's differentially expressed, we do a pretty damn good job of recapitulating this neuronal cell identity. And so this is, at least for me, a real sanity check that says if we look at what's different across these cells, are these cells more similar in their expression patterns to one another than they are across cell types? And breathe a sigh of relief and say yes. But there's a really important reason why I've taken you down this rabbit hole with me. And by the way, if we use some machine learning approaches, both supervised and unsupervised, again, we really don't do well with everything expressed, but when we look at what's differentially expressed, we have a really, really high prediction accuracy for these cell types. And this is not just to feed my fever dreams. This is actually critically important, and we play a critical role in the world of single cell genomics, because you may have noticed that there is an absolute explosion of single cell transcriptional profiling going on that is geared towards trying to identify cell types in retina, in sensory organs, spinal cord neurons, you name it. Everybody is using single cell approaches in order to identify cell type. And remember that when we do so at a whole transcriptome level, we know who we have and we fail to do this. Furthermore, a huge amount of money is being invested in this effort. This just really scratches the surface. The NIH has put at least $250 billion into brain cell identification networks. The Facebook folks have decided that they want to seed a human cell atlas. And yet we have this huge limitation of if you don't already know what the cells are that you're trying to identify to employ these analyses, then how are you going to know whether you're right, whether those are not just falsely identified cell types? And so this is a huge problem because we only got to this conclusion because we worked backwards from identified cell types. If you're cutting out the basal, lateral, amygdala, whatever, and separating it into cells, you don't already know these cell types, you're only working backwards. And so I think there's a real caveat to be brought based on working backwards from known cell identity that I hope that we've started to communicate to high throughput genomics folks. That doesn't mean that there aren't tools at our disposal. So what can we do? Well, the last thing that we were able to show is that using pooled variants really helps here. And so I'll cap this off by saying, if you don't already know what your cell types are, there are approaches that can hopefully point you in the right direction. If you haven't thought about pooled variants in a while, again, pardon me for the clumsy approach. if we have 3 cell types and we're looking at the expression of lots of genes in them, then the idea behind which genes will contribute to unique cell identification is encapsulated in these graphs. So here you can see for gene A in cell type 1, we have a defined range of expression that's very similar across cell types in terms of a range, but their overall amounts are quite different. And for gene B, we have very similar ranges and their overall amounts are quite similar. With cell types identified, you can see these differences. By using, if we are blinded to cell type, then if we pool these, the variance will be much higher in the pooled populations than in the individual populations, and that might signal a gene that is differentially expressed across cells, even if you can't possibly know upfront which cell types they are. Whereas if we pool cell types 1, 2, and 3 here, the variance won't change. And so we would have a low variation expression variation gene on the right and a high variation gene on the left. Now, this is not foolproof. So here we have combined variance in gene A that is much higher than combined variance in gene B. And so here we would conclude that this may contain information discriminating among cell types. The problem, and we would exclude gene B because it's not going to have a lot of information for us. The problem comes with the expression profile in the right, where you have highly variable expression in each cell type. Here, as you pool these, you're actually going to end up with very similar variants in genes A and B, and we might inappropriately include gene B in this analysis, but we could not a priori eliminate it, and so we are going to get a little bit of low information inclusion with this analysis. But this is all to say that if you're blind to cell type and you select for the most variable genes in the population, and there are various ways to do that. So with our data set, when we select for the most highly variable gene expression, we do much, much better at recapitulating cell identity than we do relative to all expressed contigs. So when we decided on a level of high variation, we came up with 922 of the most variable genes relative to 27,000. And yeah, we've got a whole node here that is a little bit problematic. So we've got some good clustering of LP here, of PD, of VD, and GM. But we get a little bit of noise still here in the system. And I think that would be indicative of these gene Bs on the right, where they don't have much information, but they're included in a highly variable gene pool. Anyway, so lessons learned is that we have some potentially erroneous conclusions that can come from high throughput sequencing approaches, but there are analytical tools at our disposal to lead us in the right direction. You're very patient. And so finally, I just want to end this section by making a really quick pass through of both the candidate gene approach and using all STG cell types. And I'm not going to belabor this, but we've also been using multiplex PCR to look at lots of transcripts of interest simultaneously. In multiplex PCR, we can use multiple colored probes to quantify 5 transcripts from a qPCR reaction simultaneously instead of 1. And this lets us multiplex our qPCR. And so what Adam did in working with, again, Eve's lab on this group of cells that we identified and all of these target genes of interest is he did qPCR on all of these genes of interest from individual neurons. That data set consists of 11 cell types from the STG, 65 genes of each. So we get to put 8,200 values into a data matrix. And that includes some outgroups of cardiac ganglion motor neurons. And when we use this clustering approach across all cell types in the STG using a really targeted genes of interest for us, we do a really nice job of being able to identify cell types. Not perfect. You'll see that the gastric cells get a little confused here. A really nice outgroup showing up of cardiac ganglion large cells that provides a lot of peace of mind. And so combining sort of a targeted functional genes of interest and a multiplex qPCR approach can get us a long way towards understanding cell identity as well. I'm going to skip this. We can also use other methods like principal component analysis to look at this beyond hierarchical clustering and see some reasonable separation in principal component space of these cell types as well. And so what are the limitations here? I hope I've been clear on the limitations here. Our initial RNA-seq data really don't suggest, at least for STG cells, that a whole transcriptome is vastly different across these cells. This is really not that surprising in hindsight. STG neurons are probably far more similar to one another than any other cell type in these animals. And so we may have just gotten down to a level of distinction that's just not that distinguishable. What we look at differentially expressed across them, we do a really good job for unique cell type expression patterns. And that provides a cautionary tale where using single cell transcriptomics without any a priori information has some potential drawbacks to it. And then finally, and I'm going to take my last 10 minutes here to get to probably what you might be most interested in, and that is what can we do with molecular biology to generate functional insights into actual STG circuit physiology. Proceed with caution, again, is about limitations here of using mRNA in a physiological context. And so what levels of exploration can we use in this context by measuring and manipulating steady state mRNAs? Well, I think that we can use the presence of a given gene or gene product in cell types as an implication for function. So using mRNA levels to have an implication for whether a gene is exerting a function in a cell or not. Exploring possible co-regulation or interaction of gene products by looking at correlations and correlated mRNA relationships. Combining mRNA and known physiological parameters to make inferences. And then finally, again, the gold standard here would be manipulating mRNA levels to make more direct assessments of physiology. And I have brief examples of these just to give you an idea of what I'm talking about. So the first would be simply using the presence and relative abundance of various genes in single cells to make an inference about whether that gene product might be influencing outcome and physiology in STG. So what this is the same data set I showed you from Adam Northcutt's qPCR across all cell types, but we've just pooled cell type and looked at all of the ion channels that we identified to ask which channels are present in STG cells and which might be less influencing functions. So really low abundance transcripts like these are probably, if we want to make an inference in that regard, not having as much of an influence as some of the higher expression level transcripts. And with this, we can ask, are there membrane conductances in STG cells that we might be overlooking through limitations of pharmacology or being able to separate these? I have a few candidates that I'd love a real physiologist to look at. So for example, this SKKCA, calcium activated potassium, is something that I've not seen looked at in STG cells due to my understanding is largely a failure of apomin to be appropriate in crustaceans. We see this not only in relatively high abundance across STG cells, but forming some interesting correlation relationships. And so I'd love to know more about what SK might be doing in our cells. To my knowledge, nobody has looked at the EAG-like potassium channels, the ether-agogo-like channels in STG cells, and yet they show up as fairly highly expressed. And then I know we all look at leak, but the 2-pore K-leak channels are also pretty well represented in these cells. And so just using abundance to ask what else might be in there that's exerting a function is one way to make inferences with molecular biology data. Oh, there we go. That one's slow. This is the most apology I can possibly give for a slide, and the NJIT folks have already seen this. Instead of looking at those data pooled we can look at those data across every individual cell type in the STG. And so this is ion channel expression broken up by channel across cell type or by cell type using these radial plots. What we might be able to do is use relative abundance across cell types here to look for potential physiological implications. So for example, if we do want to think about that SKKCA channel, we might expect it to exert less of an influence in, for example, LP or PY, and a much larger influence in cells like LPG or IC if there's any relationship between mRNA abundance and physiology, and that would need to be experimentally determined. And we could also just look at the suite of how different cell types are using different relative abundances we can make inferences about how they're using different relative abundances based on mRNA by looking across cell types. So one possible way to use these data, and we have these data for glutamate receptors, amine receptors, you name it. And I know that that's a lot to look like, and I know, Eve, that I desperately owe you these data. And so we're working on just finishing off nicotinic receptors to get these data out to you. Going back to Veronica, I would say that she also used this same kind of approach. So I would just remind you of the inference that can be made by combining known functional data with qPCR amounts. So I'm not going to go over this again, but the same general idea of combining the relative abundance or presence of a given mRNA in a cell with known physiology to implicate that gene in cell function. We could take a look at, again, a paper from Deb Barrow and Ron Harris-Warwick's lab to really now start to equate mRNA abundance and membrane conductance. And so I have to pause on this slide for a moment beyond just explaining to you what Deb did, but I'll explain this relatively quickly. What Deb did with the Schall transcript is she looked at IA in all of the cell types, these 6 cell types, sorry, of Pannularis, and even expressed these ectopically to look at what the A-type current looked like for Schall and Shaker. She then designed, and keep in mind this is back in the mid-90s, a single cell quantitative RT-PCR assay that used the incorporation of and densitometry of gel bands in order to quantify this, came up with a beautiful standard curve for being able to look at absolute copy number using this approach. She then looked at the mean levels of Gmax across cell types in Pannularis pyloric cells and the number of Schall transcripts to look at whether sort of on a cell population level, whether overall abundance mirrors membrane conductance. And then really, really lovely correlated the population level Schall transcript number and the measurements of IA in these cells to truly implicate copy number of Schall and A-type current in these cells. And the reason why I have to pause on this, and you'll pardon me if I get a little bit sort of philosophical here, this paper, and I don't know if Deb's out there, but I know Ron is, this paper literally changed my life. It came out about mid-graduate school for me. And I was so taken by this paper, the idea that you could combine electrophysiology and single cell molecular biology. And keep in mind, this was not a time when single cell molecular biology was something that you could do off the shelf. I mean, Ron and Deb absolutely pioneered this. I read this paper and it changed my trajectory long-term from behavioral ecology to circuit physiology and STGology. This is the paper that drew me to Ron's lab initially as a postdoc. And it literally changed my entire scientific career. And so thank you for that, Ron. That was really awesome work. And I get a little emotional, quite frankly, because you have those pivotal moments in your career and we live this stuff, right? So, and if Deb, if you're out there, your work sort of changed my scientific career. So I had to put this in there and put that in there because I don't think we give each other enough credit for the influence that we have on our careers. Anyway, so I loved this paper and I thought that this was a beautiful contribution to how molecular biology can start to look at physiological implications. I don't teach anymore, so my teaching voice is not in conditioning. And then finally, another way to think about this is to look at whether correlations among mRNAs actually suggest potential co-regulation and functional relationships among gene products. And this is something where really I was able to, I think, make a contribution with Yves when I was a postdoc, and Jean-Marc was pivotal in starting this out as well, where we took single cell qPCR measurements of a bunch of different ion channels and identified cell types, and somehow really fixated on their correlated relative abundance to one another. So each point is the quantitation in a given cell. Each color is a different cell type. And we saw a bunch of cell type specific correlations among channel mRNAs. And I've gone on to do a lot of work using this same sort of perspective. It's burned into my brain to look at mRNA correlations. And so Simone has followed this up. Everybody in my lab has basically followed this up in one way or another. And again, we see these patterns of correlated ion channel transcript numbers. But I thought one of the most satisfying chances that I got to work with a real membrane biophysicist was in working with Jorge on this project as well. And so Jorge and I teamed up to essentially in the same, not the same cells, but similar populations of cells, look at the correlations among A and HTK and H conductances in PD neurons, for example, and the channel mRNAs that underlie them. I found it very satisfying that we were able to identify the same correlations at the mRNA level that Jorge's group was able to identify using voltage clamp. And so again, while this is not absolute evidence that all channel mRNAs are indicative of conductance, this is, again, really strong evidence that there is a physiological underpinning to some of the expression patterns that we're seeing. This is an independent data set generated through a different study of Simone's, actually. This doesn't appear in the paper because we for some reason, didn't publish the H results in that paper. But she was able to beautifully recapitulate those mRNA correlations as well. And that repeatability is something that I think is really important. And then finally, it is possible to directly compare mRNA levels and physiological parameters. And this is the paper that Dirk was alluding to in the introduction, where Jean-Marc and I, working in Yves' lab, teamed up to take his mastery of membrane conductance in biophysics, and he conducted all of the voltage clamp measurements of, for example, IA, delayed rectifier, and calcium activated potassium in a group of PD neurons. What is distinct in relative to the studies you've seen previously in the previous slides is that we did the qPCR and the biophysics and the membrane conductance measurements in the exact same neurons. And find really, I think, compelling correlations that suggest that mRNA copy number is indicative of membrane conductance. And then, I don't know if Joey was able to join us today, but I dug this one out of a cardiac ganglion data set that Joey generated as well, looking at calcium currents in cardiac ganglion motor neurons and copy numbers of one of the voltage-gated calcium channels. This is an inward current, and we've left that as per the nomenclature. So this is not a negative correlation. This is a correlation with inward current and calcium channel expression. I will say that we've had various goes at this and never 100% could recapitulate this to publish it, although I think that part of that is due to how difficult it is to measure calcium channels in these, calcium currents in these damn cells. Daniel and others can attest to that. And so real, I think, direct evidence that using mRNA as a physiological correlate has some validity to it. But again, I promised you limitations. And so the major limitation here, of course, is that steady state mRNA, it turns out, isn't protein. Who would have thunk? And so the transcriptome is far less complex than the mature proteome because of all the post-translational modifications that occur, because of all the translational regulation that can occur. And so a tremendous amount can occur between when we measure steady state mRNA and any of the endpoint physiology measurements that you folks are making. All of these post-translational modifications will not be represented in any mRNA analysis. Interactions, protein complexes will not be represented. And so you always have to take with a grain of salt what mRNA abundance can tell you about what the protein is doing and what that protein is doing to the cell. And so it's really these comprehensive approaches that STG can bring that I think make us stand out in the community of truly understanding sort of single cell circuit dynamics from the genome up to the full circuit. Finally, the gold standard would be manipulating mRNA levels to demonstrate that there is a real impact on physiology. And I have to go again back to Ron's lab through work that Jason McLean did. This was followed up beautifully by Jason and Marie as well in follow-up studies, where injecting shell mRNA into PD neurons in pannularis produces this really obvious substantial increase in the A-type current. And if I recall correctly, though, Jason and Ron were having some difficulty understanding why they were not moving the function or the physiology of these cells. So the shell-injected PD neurons were really difficult to distinguish from control neurons. It was really only when they looked at other currents that they realized that there might be a compensatory increase in IH that was helping to balance the increase in IA. And again, I think one of those presented in hindsight sort of perspectives, Jorge and his lab found a real strong relationship, at least in Cancer borealis, between A and H conductance in Cancer borealis. And we found this also at the mRNA level. And so when you take Jason and Marie's work from Ron's lab and you combine it with what we've seen with Jorge and my work, I think we really start to see how mRNAs and messing around with mRNAs can really be recapitulated at the physiological level. So another beautiful study coming out of Ron's lab that really set the standard for molecular physiology in the STG. And so I'm done, you can exhale. And so where I've taken you through probably a little bit of a long droning approach is to start to ask questions then from a real sort of why would we want to focus on this kind of approach? And so I focus on this because ultimately for me, every cellular process originates at the level of expression of relevant gene products. And the power of these approaches is they really let us look at many genes simultaneously, or even with the advent of high throughput sequencing, all genes simultaneously are gene products. And we can apply a common methodology across questions in model organisms and levels of analysis, because there is really a profound similarity at the genomic level that we can really start to apply comparative approaches here. And so what molecular approaches allow us to do is really provide a snapshot of information of the gene expression state of a cell or a tissue at a given physiological or behavioral state. But that snapshot is also one of the limitations. And so pros and cons, we really have prolific amounts of data that can be collected. We can easily archive that and make that publicly available. The NIH has invested in huge infrastructures that I showed you to share molecular data. Data are truly universal. A, T, G, and C works no matter, for the most part, there are small exceptions to this, for any organisms you want to think about on this planet. And we can use powerful amplification techniques to look at a very fine scale, single cells or smaller, single nuclei, mitochondria, et cetera. But there are plenty of limitations to molecular approaches. That signal of measuring RNA is quite far away from that final functional product. So post-translationally modified, trafficked, and targeted proteins and RNA are worlds apart inside the cell. And with very few exceptions, all measurements are terminal and completely lack dynamics. When we pull a cell out for molecular analysis, that's our one shot. We do not get to measure shell over 72 hours or a week in culture. not at least not very easily, and we certainly can't do it. And these detection processes are based on biochemistry. So while I feel like even though there are tons of vagaries in electrophysiology, that is at its core physics, and physics is, I think, hopefully relatively reliable from that perspective. Biochemical processes are subject to the vagaries of the stochasticity inherent in the system. Primers finding templates, temperatures, enzyme efficiencies, batches of enzymes from manufacturer A versus manufacturer B. And so I've sort of, become fond of this phrase we use around the lab, that a lot of the work we do is relatively absolute. I would never swear to you that I have accurately measured 1,000 copies of an mRNA in PD, but I would base a lot on the fact that if there's 1,000 in one PD cell in our assay and 2,000 in another, that we're in the ballpark and they really are twofold different. And so there are limitations to the absolute nature of this. And finally, I like to think about targeted gene approaches because, as you've noticed, I'm not all that smart, and this embarrassment of riches, I don't know what to do with 10,000 genes. I'm lost in thinking about that at that scale. Give me a dozen ion channels to think about how they might work together, and I might be able to figure it out. And I think sometimes we generate data to generate data with the hopes that somebody down the road knows what to do with it. But there really is an embarrassment of riches here, where data is being generated faster than anybody can possibly analyze or comprehend it fully. So with that, I'm gonna not go through a long list of thank yous. I apologize if anybody who should be on this list isn't. I wanna thank you to everybody that I've worked with in the lab, previous and current, with an incredibly talented group of folks that have made this all go. Without the collaborations that I've mentioned here and beyond, I'd be nowhere. And so thank you particularly to my chief collaborator in this, Eve. We've done a tremendous amount of this work together. And that's been sort of one of the most satisfying relationships I've had in my scientific life, to say the least. We've been generous to be funded through multiple mechanisms for this work. And I don't know if there's time or energy for questions, but I'll do what I can in the time that's left. So thank you all very much. I, again, appreciate the chance to share some of this with you.
Thank you, Dave. So it's my honor today to do some moderating. I think by now you all know how this works. So just keep shooting questions at Dave and start discussing. But if you want to, feel free to use the raise hand feature if you don't want to brazenly interrupt whoever is talking. If you're using an older version of Zoom, you find the raise hand feature in the participants list. With the newer versions, you find it under the reaction tabs, which you can use to clap your hands as well. If you don't find any of these, just type question mark in the chat. So I'll keep an eye on the chat and on the raised hands, and please start discussing. Dirk has a question. Yes, please, go ahead. And please unmute yourself.
Dave, I don't know. I may have asked you this before, but I don't remember your answer. So how do you imagine, what strategies could there be to look at things where there isn't a relatively close physiological readout of expression? So ion channels, of course, you can look at expression and you can measure the currents. That's not a direct relationship either, but it's relatively close. Receptors, it depends. But then you go through things like, I don't know, transcription factors or whatever, where it's not clear where that approach that you want to take, like what would you actually measure to, I mean, things become more and more indirect, and you have no confidence or no good way of testing that I can think of if these things are actually related to each other.
So yeah, I think that the short answer to that is that we get in these gene regulatory pathways that are the domains of biochemists that are far beyond me. What I would start with is, and if you gave me a, sort of a, remove the technical limitations, I would start with conditional expression and conditional repressor systems. So conditional knockdown, conditional activation of various genes, targeted gene editing in ways that would allow us to hopefully dissect upstream elements and then use what the STG is good at, which is downstream readouts. So maybe if we could construct a series of experiments where first we do something that is a manipulation that we know changes that readout. I would then do something along the lines of high throughput sequencing that would allow us to look at gene regulatory networks. So generate sort of a systems biology approach to looking at expression in that cell. And then try our best to identify those pathways and then use these conditional systems, RNAi, inducible expression systems, et cetera, to try to tease that apart. So the reason why I go to conditional and sort of activatable systems is one of the things I've loved working on as an STGologist is compensation in various ways. And while I am certainly one to acknowledge the incredible breakthroughs that knockouts and sort of long-term knockdown have taught us, I also worry that those negative phenotypes that the knockout folks see is simply a long-term developmental compensation, and they're ruling out what gene products might be involved in a process because of these exquisitely beautiful compensatory mechanisms that exist. So, so that's my long, short answer, is that if I could get in there and sort of turn on and turn off or functionally activate or functionally repress various gene products along a gene regulatory network using STG as a readout, that's what I would try to do. Or hey.
This may be a self-evident question or the answer to it, but when you extract the RNA of a cell, you are making a mess of that poor cell, perturbing it and all kinds of things. So it makes you at least question how faithful that RNA expression levels, correlations or the lack thereof, etc., are. So I'm not, I mean, I acknowledge that this is all true and whatever, but is there anything that you know that one can do to minimize that? I mean, is there a little laser that you can just shoot at the cell and instantaneously evaluate everything? I mean, that would be the ideal, but I mean, this now brings into question a lot of consequences, functional consequences that you can or cannot actually derive out of these measurements, except for some obvious or more clear ones, right?
Absolutely.
So where do you take it from here?
So there's a couple ways that I would think about that. One, and I'll do that annoying thing where I reword your question a little bit too. When you're looking at mRNA, you're also worried about a time lag of what does that transcript or transcriptome look like relative to the instantaneous physiology of that cell, and is it predicting what that cell will be doing 4 or 8 or 10 hours from now, or is it lagging behind what that cell is doing? And those are all incredibly valid concerns. The two ways that I would address that technically would be, one, even though it sucks and it mostly doesn't work, you can collect RNA from fixed cells, and you can fix cells relatively rapidly. So one of the, I guess even though mRNA is finicky, the processes that change mRNA levels relative to what we think about at a physiology level are glacially slow, right? So membrane conductance changes on a millisecond level. It takes hours for changes in mRNA to show up after a perturbation in most characterized systems. So I take some solace in that fact. And then we do probably too much sort of methodological validation in my lab, and I apologize that it all sort of stays in my head and stays on our shelves, but we've done a whole bunch of analyses for STG specifically where, there's a protease digestion associated with our cell type collection. Well, we've done that protease digestion with ongoing physiology recordings, and we don't perturb the rhythm with it, so that makes me pretty happy. We then from there try to very rapidly go into what Eve sort of pioneered with her single cell collections for transmitter identification, which is this sort of replacing the physiological saline with ethylene glycol and such. This is a pretty rapid changeover for us. And I feel like in that point, we're basically eliminating biochemical activity, which would change these RNAs. So I think we're doing reasonably well there. And then we've done all sorts of time series of, TTX exposure, TEA exposure, all the things that we can possibly think of that drastically screw up what these cells do. Because I'm interested in activity-dependent feedback. And really have never seen any detectable change in the RNA within an hour or 2 hours. And so through that and the sum total of a whole bunch of other gobbledygook that I don't have time to go into, I feel very comfortable in the collection protocol that we're doing for these single cells, that it is at least a snapshot of that cell within that time period. But biologically, I cannot tell you what that cell looked like when it was in the animal and what taking it out of the animal might have done, the gross dissection, et cetera. Here, I think we have to rely on sort of the sum total of the inferential knowledge of the fact that, well, we can correlate transcript and membrane conductance. And we can see that in Panularis and Cancer borealis and across diff. So I feel like it's not just the Wild West out there, that we're doing a reasonably good job, but all of those concerns are always at the back of my mind.
Thanks. Yeah.
Ron?
It was a really wonderful talk, Dave. Congratulations. And so carefully explained that all of us can understand it. It was really, really a great job. One of the things that impressed us when we were just getting started on this about getting the genes for the ion channels, it immediately became clear that when you're measuring one of these genes like SHAL or Shaker, the question is, which one are you measuring? We found enormous amounts of alternate splicing of the RNA, dozens of variants, and for any one of those genes that could absolutely dramatically alter the properties of the resulting current so that it can switch from a very rapidly inactivating A-type current to a very slowly activating, non-inactivating delayed rectifier current. The voltage dependence for activation could shift by 20 to 30 millivolts in activation and activation. So you can get entirely different functional channels from the same gene. Now, presumably all your measurements are made from one sequence, which presumably is in the common sequence that would measure all those splice variants. But I'm wondering if the specificity of the RNA-seq at the single cell level would allow one to start looking for sequences specifically in some of the splice variants that are separate from one another. And you could find out, for example, whether, all of them seem to be expressed because we pulled them out, from the stomatogastric ganglion, but some of them might represent 90% of the total and others 1%, in which case we could, essentially ignore it. Could you address that question?
Sure, I will do my best. So I'll answer that sort of in waves. We do specifically design our analyses, and these have been around for a while, so I bet the people in my lab now don't even know this, to target the common aspects of the sequence as much as we can. So we are trying to capture all splice variants in the analyses that we do. And I think you've hit on a point as to maybe this is one of the reasons why it can be quite challenging to directly correlate mRNA and membrane conductance, because of these cell type specific differences in alternative splicing that we see, that you've shown, that we've shown as well in sodium channel, for example. And I actually think that the seq approach might not be well suited to grab this, because unless you get very lucky where that fragmentation happens, I think it would be very challenging for the mapping algorithms to be able to identify a given splice variant. And that's because the fragmentation in the RNA-seq approach is our worst enemy in this regard, because as you're alternatively adding exons and such, then it might not be that there's overall a difference abundance of the exons in the whole cell, but you really need the whole splice variant to be able to in order to be able to do that. And so that's where I think we'd run into a lot of trouble trying to measure splice variance and correlate that with membrane conductance. What I would rather do is, and this is probably because I try to make things as complicated as possible whenever I can, is I would target multiple sort of overlapping probes that would give us an unambiguous readout of what the splice variance would be present in a given cell type. And then be able to use that to build back, sort of a relative abundance of splice variance. So as I'm thinking about this out loud for the first time, if you maybe, for example, had a probe that detected every exon, then you might be able to look at the relative abundance of exons and build back from that the likely relative abundance of splice variance, right? I don't know if that would work. I'd have to think through that. And also what we're all ignoring here that you've been kind not to bring up are things like RNA editing events, right? So even if we get exactly the same quantitation, it's been well demonstrated that a single nucleotide change can have profound effects on the protein, and that happens during the post-transcriptional process. So RNA editing is a huge thorn in the side here as well. So that long meandering answer is designed to distract you from the fact that we haven't done this, that I can't think of a good way to do this without going to a tremendous amount of effort, and ultimately would want to really define the question that would get it, that approach would get at in order to tackle that. But now I want to do it because, and so sorry to the folks in the lab for me now designing an overly complex set of overlapping probes and such, but I think that would be a really fascinating take on it. So we don't have anything, I don't have anything smart to say about that other than we're looking at pooled splice variants intentionally at this point. Hi, Dawn.
Hi. Dave, I was wondering about how strong the warning should be to folks that don't have identified cell types. So do you think if, as long as they're looking at neurons, the high variability is going to be close to what you see, or is it because we're looking at rhythmic neurons that it comes out pretty good? I'm just wondering what kind of warning should go like across different types of sensory versus motor neurons, et cetera?
Yeah, that's a great question. It's not as though we're the only ones thinking about this. There's certainly a cautious group of folks out there who have brought up these kinds of caveats. And I think that the caveats are usually included in most of these studies. But there is a tremendous amount of, I think, pressure to basically publish these studies and move on. And so I'm not entirely convinced that the caveats will ultimately outweigh the brute force that's taking place. But I have a tremendous amount of hope in this regard as well, because I think that what is happening is that there is an explosion of technology that is unfortunately making me a little bit uneasy as an STGologist of being able to combine high throughput patch clamp with high throughput RNA-seq and genomics in a way that really will now take a multimodal approach to understanding cell type in these tissues. There was a beautiful paper in Nature this past year that I'm still trying to sort of wrap my head around in terms of the combination of high throughput patch-seq and high throughput sequencing that I think is honestly a game changer. And so I do think that by and large, the cell identification community understands that multimodal information is the best path forward, but are willing to generate huge data sets in the meantime because the technology is available and then move on from them. So I don't know, that's a crappy answer. Eve, what do you think? Can I ask you, put you on the spot, what you think the identification folks, how the caveats are coming across and whether that's an issue or not? Because you and I have talked about this a lot.
Yeah, I actually think that the most important other piece of information that the cell type people need is projection patterns. Because if you combine, if you think about the way we identify cells, we identify them on the basis of their projection patterns primarily, and then pieces of their electrophysiology. So I actually think that as the connectome work progresses and as people start looking at these long-term projections, some of the cell type ambiguity that comes just from doing the molecular work is just going to dissolve, because you're going to see that these cells go from here to here and those cells go from there to there. And it's hard to say that it's the same cell type when they're going to totally different areas of the brain. So I think in 10 years, this problem will have been resolved with a combination of the anatomical connectome stuff and the molecular work, which will end up with, and I actually think the projection patterns are the most reliable single way of determining cell identity. I mean, the electrophysiology per se is very changeable. We know that, right? And the molecular stuff is going to be somewhat changeable. The projection patterns, yes, can change, but they're much less likely to change than the other two. And obviously what you would really want is all three, right? You'd want the electrophysiology and the molecular characterization and the projection patterns. But if I had to pick any one in addition to the molecular work, it would be the projection pattern. And I think that that's going to come with the connecting stuff.
Yeah, I agree. I like that 10-year number too. And so I think that, you know, there's going to be such an explosion of connectomics.
Yeah, I'm not a very patient person, but I think that I think we're going to be looking at the next 5 years of single cell molecular garbage, and then another 5 years of single cell sort of connectome partial answers, and eventually there'll be a convergence as those two technologies end up being woven together.
I'm going to be annoying and share my screen. This is the paper I was talking about. I'm still trying to sort of wrap my head around this paper. I really like it. I mean, it certainly has limitations, but you know, they did combined patch clamp, biocytin staining, and single cell seq for 1,300 neurons in mouse primary motor cortex, and came up with this, you know, kind of really beautiful analysis of electrophysiological phenotypes and layers and molecular biology and such. And so I hope that we'll see more studies like this that really start to push the envelope of multimodal
Go back
information.
up to that figure where you see the anatomy as well.
What's that, Eve?
You go back to the figure you just showed too quickly. You'll notice they had the projection patterns of the anatomy is actually really, really important, because
Absolutely.
it really distinguishes as you start, you know, some of them are going from layer 1 to layer 5, others are going from 2 to 3 or whatever it is. And you're really hard pressed to say that even if cells have very similar molecular expression patterns, if they're going totally different layer projections. And of course, it's really important to remember that when you cut slices, you actually cut through the long-term projections. And Nelson Spruceton, who's not, you know, has done some very pretty work with these long-term projection patterns that show that even two cortical cells that sit next to each other can be going to totally different parts of the brain. So I think there's the local projection patterns and then the long-range projection patterns that it's going to require people doing a lot of very difficult, I think, light level work on many neurons to get those long-term projections, which are going to be crucial in knowing, you know, this cell takes information from olfactory cortex to motor cortex, and this one goes from olfactory cortex to somatosensory cortex or whatever, you know.
A lot of work's being done in the spinal cord that does just that, where they're doing single cell RNA-seq combined with retroviral measurements, so that they're only studying the cells that either receive or give input to one other cell type. And combine that with their anatomy and their physiology. As you say, it's heroically large amount of effort, but it's starting to yield interpretable results, whereas otherwise it's just all over the map.
Yeah, I mean, that's what has to be done, right? Because then you get it right. At least approximately right. So we really need the old-fashioned anatomists, except they're now the new-fashioned anatomists.
It's old as new again.
Yeah. What goes around, comes around. Anyway.
Well,
It was a really beautiful talk.
thank you.
We are indebted to you, but I think probably we all have to go away now.
Yep, you've given me 90 minutes of your lives, and I greatly appreciate that. So thank you, everybody who came today. It's always a pleasure to talk to this group, and I look forward to the next one.
And it's recorded.
Thank you. Before everybody leaves, Smita and Nelly, do you want to say anything about the next talks coming up?
Next, yeah, go ahead. Nelly.
It will be on the February 26th at 2:30, as always, Eastern time.
Who?
Oh, hi.
Okay, and I'm waiting to see the next woman talk.
Yes, it's coming.
Don't forget.
Okay, thank you. Bye, everybody.
Bye-bye.
Thank you, everyone. Bye-bye.
Bye,
Thanks,
Dave.
Dave. That was great.